right. So welcome everyone to the third roundtable of 2026. It's my great pleasure today to introduce Dr. Sumita Nandadasa. He is an assistant professor in the Department of Molecular Cell and Cancer Biology and the Department of Pediatrics at the University of Massachusetts Chan Medical School. His research focuses on the molecular mechanisms that regulate primary cilia assembly and functioning during development and disease. His laboratory has uncovered important roles for ADAMTS metalloproteinases and ciliogenesis, revealing unexpected intracellular functions for these proteins in regulating ciliary structure and signaling. Through genetic and mechanistic studies, his work is helping to define how disruptions in cilia formation contribute to ciliopathies and congenital disorders, providing new insights relevant to diseases such as polycystic kidney disease. So please welcome Dr. Sumita Nandadasa.
Thank you, Pam, for the introduction, and Terry and Brad for the invitation to present our work. Let me see if I can share my screen. Okay, there we go. I assume you can see my screen?
Yes.
Okay. So as Pam mentioned, I'm a relatively new faculty member at UMass Chan Medical School. I'm into my fifth year this year, actually. And my lab studies a number of other organs. I'm going to mainly focus on the kidney today for this audience, but pretty much all the organs in our body are affected by the molecules that I'm going to talk to you today, which happens to be a bona fide extracellular matrix molecule known as NMDS9, and also the ciliary transition molecule TMM67. So I'm going to start off with this piece of art before I get into science. It's a work of art known as Lunch with a Helmet by a Japanese artist, and it's in display in the Metropolitan Museum of Art in New York City. And this reminds me of the molecules that we're working with and how our perception of them are when we first started working with them. So the first molecule I'm going to talk to you about is ADAMTS9, and this is a bona fide extracellular matrix molecule. And when I first started working on this molecule as a postdoc, this had nothing to do with cilia or cystic kidney disease. And the other molecule is TMM67. This is a bona fide ciliary transition zone molecule. But it also has a very important role in regulating Wnt signaling in our tissues. And it wasn't clear how these molecules act together or how they're regulated embryonic development. I'll come back to this piece of art at the end of my presentation and give you a little bit more insight on how this looks like a motorbike at the very end. So I don't think I need to introduce this organelle to this audience. As you know, the primary cilia is a key cellular organelle present in nearly all the cell types in our body. And when you think of ciliopathies, the easiest way to think of ciliopathies is a scenario where there's a cilium and no cilium. But as you know, it's not as simple as this. There are all these other conditions where if you lose these cell surface receptors, for example, that's present on the ciliary membrane, you can get ciliopathy such as PKD. And also if you lose specific kinases in our body or phosphatases, you can get these extremely long or short cilia. And also molecules that are involved in trafficking these GPCRs and other cell signaling molecules can also, loss of these molecules can also cause abnormal cilia. There's a whole other class where motile cilia are affected, and you can have PCD by the loss of ciliary motility. Now, depending on who you ask, there are about 300 to 400 genes that are thought to be involved in cilia formation and cilia function. But nearly all of them are thought to be from the intracellular compartment. What I am interested in is the extracellular compartment, because the molecule that I started studying as a postdoc happened to be in the extracellular compartment. But I should point out, this is, if not the only organelle in our cells, that are nearly 90% outside of the cell. So the extracellular compartment is very important. And let me show you this beautiful EM work done by C.A. Poole looking at chondrocytes. And what he showed is that primary cilia are not just extending into the extracellular microenvironment, it is actually intricately interacting with some of the extracellular molecules. And where he pointed these arrows and arrowheads, that's where the ciliary membrane is interacting with collagen and proteoglycans. This is published way a long time ago, and he also showed that if you have a lot of extracellular matrix, it can actually bend the cilium, and if you um, remove the extracellular matrix by digesting with trypsin, the ciliary membrane also responds, and you can have a completely straight cilium as well. So this is the main question that my lab is interested in investigating. And what I'm going to talk to you today are extracellular matrix metalloproteinases. These are proteins that are involved in remodeling the extracellular microenvironment in our cells. There are three major classes of metalloproteinases, and I'm going to mainly talk about ADAMTSs. Now, more often than not, people misrecognize the ADAMTSs with ADAMs, but I should point out this is a complete different family to ADAMs. ADAMs are 100% of the time transmembrane molecules. ADAMTSs are secreted molecules. They completely act outside of this the cell. And MMPs, on the other hand, can be either transmembrane or secreted. And I'm going to touch upon MMPs a little bit too towards the end of my talk. So why are we interested in ADAMTSs, and what does this have to do with cystic kidney development? So during my postdoctoral work, Friedhelm Hildebrandt's lab, and we collaborated, and they identified the very first human patients known for ADAMTS9 mutation. There were two families, and both families had NPHP, and the second family, in addition to NPHP, also had Joubert syndrome. And just to remind you, what's unique about NPHP compared to ADPKD is that these make highly cystic medullary cystic kidneys, but they do not expand similar to ADPKD. They're either small or same size as a healthy kidney. So it's not really clear why that is the case and why these kidneys do not really expand. But one thing that's known is that these kidneys are also highly fibrotic. Now, since 2019, when the very first patients were identified, now we know 6 additional alleles. So you can see the number of patients are increasing by every year, and there are 8 published ciliopathy patients for ADAMTS9. So it's a very important enzyme that's required for NPHP and Joubert syndrome. There's a 9th allele. This is unpublished. This is by personal communications. This particular patient, boy, has both NPHP and Joubert syndrome. And all of these patients are recessive homozygote mutations. So these are loss of function mutations. So just to give you a little bit more information on the ADAMTS family and where ADAMTS9 is. ADAMTS9 is this particular molecule in mammals and most vertebrates. It is duplicated, so there is an identical sister protease known as ADAMTS20. In non-vertebrates, there's only one of these, so it's and that gene is referred to as GON1 because it was first identified in C. elegans, and the worms have gonatogenesis defects. This entire family is believed to be evolved from ADAMTS9, so they're all evolutionary products by gene duplication. And this enzyme family is categorized based on the extracellular matrix molecules that are known to be cleaved by these. And ADAMTS9 and 20, the canonical function of this was to cut chondroitin sulfate proteoglycan known as Versican. And it cuts Versican in this glue-ala bond at 441 and 442. That was the field when I first started working on this molecule. And we created the first mutation or the a hypomorphic mutant of ADAMTS9, the complete null of this enzyme dies at gastrulation. This is a hypomorphic version of ADAMTS9, and these are dual. And what we found out from the mouse work. So essentially, this enzyme is secreted out of the cell as a zymogen. So there's a pro domain that is associated, and once it reaches the cell surface, furin activates this molecule by removing this pro domain. And the canonical function of this molecule, as I mentioned, is to cut chondroitin sulfate proteoglycans. And what we found out was that part of this enzyme is recycled back inside the cell through LRP1 and LRP2 mediated endocytosis, and is trafficked to the recycling endosome and is in the lumen of the vesicles that give rise to the primary cilium. So it's in contact with the cargo molecules that are part of the primary cilia. At time of publication, what we didn't know was what the substrate of this enzyme is that is required for ciliogenesis. And towards the end of my postdoctoral work and the onset of my own lab, we did a very specific proteomics analysis, try to figure out what this molecule cuts that is so essential for ciliogenesis. This technique has a very long name. We'll simply refer to it as N-terminomics for now. But let me walk you through how this technique works. So every protein has a natural N-terminus and a C-terminus. And when a protease cuts a protein, this creates a new N-terminus. And you can use this biology to identify where proteases cut molecules. And what we do, once we lyse a particular sample, you use isobaric tags that very specifically label the N-termini of proteins. And this will label the natural N-terminus and also this neo N-termini that are being created by protease cleavage. And then we digest with trypsin, which creates hundreds of fragments of peptides after cleaving, after lysine residue. And these now have unlabeled N-termini. And what we do is use a very specific polymer that binds up these N-termini, and we get rid of all these peptides that we're not interested in looking at. And we simplify the sample and run LCMS-MS. So that's the basic principle behind this. And depending on how you set up your experiment and what samples you compare, for example, if you compare a wild type and ADAMTS9 knockout experiment, this particular neo N-terminal peptide, this internal peptide, would be absent from the sample. So this is easier said than done. It's like trying to identify a needle in a haystack. The only difference is we don't look for the needle in this case. We get rid of the haystack looking for these needles. This is a little bit more detailed illustration of this, but for lack of time, I'll just move on. But essentially, we're trying to detect these internal peptides that are labeled. If you follow this process, that can be identified through mass spec. So essentially, a peptide that's coming from natural N-terminus would have labeled N-terminal peptides, both in the wild type and the mutant. For internal peptide in a protease knockout sample, you would only see it from the wild type sample. So that's the principle behind this technique, and it's a really nice method of identifying protease substrates. And this technique has revolutionized the metalloproteinase field, actually. So we used this technique, and this is what we got when we compared ADAMTS9 knockout RPE1 cells and wild type RPE1 cells. D12 is just a clone number for the ADAMTS9 knockout. And there was only one ciliator substrate, and that's the circle that's labeled by green, TMM67. I will mainly talk about that, but I will come back to the red data as well in this Volcanon plot, which is MT1 MMP, which we first characterized. And we didn't realize how important that particular cleavage was for this story as well. All right, so this is where ADAMTS9 cuts TMM67. This is the peptide that we identified. It is labeled at this phenylalanine 332 and creates this peptide, which is either completely absent in our proteomics experiments or present at very low abundance. I don't need to introduce TMM67. I think it's a very well-known ciliary transition zone molecule. It's main reason or the cause of MKS in humans, which is the most severe form of ciliopathy. And this is where MTS9 cuts. So this peptide comes from this linker region that's present in between the cysteine-rich domain and the beta sheath-rich domain. And there are two cleavage sites, and we published this last year. Now, what's really unique is that this particular linker region is highly conserved in mammals. This is quite unusual for a linker region because it's just connecting two domains. But both cleavage sites are highly conserved, and there are three mutations that are surrounding the cleavage site or within the cleavage residues itself that are known to cause various ciliopathies, including cystic kidney disease. So the first thing we did was to characterize the cleavage or validate it. So this is the TMM67 domain structure, and I've marked the two cleavage sites by red and blue here. We've now labeled a linker 1 and a linker 2 region based on the cryo-EM structure that got published on this molecule. And the proteomics data predicted that there would be a 34 kilodalton N-terminal fragment that would be released. So that's exactly what we looked for when we first started working on this. These are our wild type and ADAMTS9 knockout RPE1 cell line, the D12 line, and in the conditioned medium, we do not detect this 34 kilodalton fragment in the ADAMTS9 knockout, and you see increased full-length protein. I'm just going to use the bottom panels and just to summarize what we discovered. So essentially in wild type RP1 cells, these are ice-cold methanol fixed basal bodies or transition zones that we're looking at TMM67 in red. And the N-terminus is not localized to the transition zone, but the C-terminus is. In ADAMTS9 knockout cells, neither the N-terminus or the C-terminus is localized to the transition zone. And what we went on to show was that if you lose TMM67, you not only lose just TMM67, but you lose these 6 other molecules as well. And these 6 molecules are lost in commonly with ADAMTS9 knockouts as well. So it's a very unique subset of the transition zone that is lost by loss of TMM67, and we refer to this as the MKSB9 module. So the next experiment we did was to figure out what this cleavage does to ciliogenesis. So we generated these cleavage products. So for example, TMM67 delta 342 refers to the C-terminal half that is generated by ADAMTS9 mediated cleavage. This molecule was cloned with a signal peptide onto it, so it has the correct topology of this molecule, of this fragment, which I have not shown here. And the N-terminal fragment is the N331, and these 3 molecules on the right are either dual cleavage site mutants or single cleavage site mutants. And we simply did rescue experiments. So if you knock out TMM67 in RP1 cells, this is a cell line produced by the Johnson lab. And if you transfect full-length TMM67, you can rescue ciliogenesis, which is quantified here. And if you simply transfect the C-terminal half, you can rescue ciliogenesis to the same level. But neither the N-terminus or the cleavage site mutants can rescue ciliogenesis. So this told us the C-terminal fragment is necessary and sufficient for ciliogenesis. And we also characterized what happens to the 6 molecules that I previously mentioned. not only the C-terminal half can rescue TMM67 knockout in terms of transition zone assembly, it can also rescue MTS9 as well. We characterized the three patient variants that I mentioned that are surrounding the cleavage site, and all three patient variants were defective in coelogenesis. But very importantly, these patient variants were also defective in transition zone assembly and localizing to the transition zone. So we modeled this in worms to begin with, and this is work done by Oliver Black and Karen Lang in his lab. And this cleavage site is conserved down to the worm as well. And Karen generated two specific worm lines. The first was to model the patient variant at the cleavage site. So it's a F249V phenylalanine variant. And the second one we refer to as the delta cle worm line. Both cleavage sites are mutated in this line. And long story short, these cleavage site mutant worms behave very similar or equivalent to the TMM67 null or the MKS3 null worm lines. And the transition zone assembly was also affected in these worms. We also modeled this using mice. We developed a new mouse line. So we mutated both cleavage sites, and we refer to these as the TMM67 delta cle. And these mice only survive till about P14, and they generate these highly enlarged cystic kidneys. In addition to the kidney defect, these mice also develop hydrocephaly, and it's shown here. And there are other organs that are affected as well, including the heart and the liver in these mice, which I'm not going to show much data today. The first thing we did was to look at renal cilia in these mice, both the TMM67 knockout and the new delta cle line. This is how their cysts look like using freeze fracture SEM. They have these really long cilia or short cilia, which we've depicted in two different colors. And both these cilia types were completely defective in forming this ciliary transition zone necklace, which is this green structure or the extracellular and transmembrane compartment of the transition zone. I think first coined by Jeremy Reiter, and that is completely missing in these mutant transition zones. As you can see, their extracellular compartment is completely gone. So you can also look at these really long cilia, and they have completely abnormal microtubule organization as well. So the long cilia are not normal either. Just to give you an idea of some other cilia, these are the brain ventricles. So these are motile cilia lining our ventricles. These are ependymal cilia, and they are defective in forming the transition zone as well. But different to renal cilia, these make these really crazy looking membrane bulges. You can see some of them look like these satellites, and some of them look like these, I don't know, human ears or something. So there is a complete different phenotype when you look at renal cilia versus motile ependymal cilia. We're really excited to figure out what's going on with this as well. So we think this is excessive membrane flooding in to the ciliary membrane by the defective transition zone. So this is another type of cilia. These are our retinas, or the retinas of the mice. And this is actually a really nice model to look at the transition zone necklace, because photoreceptor cilia has this really enlarged transition zone known as the connecting cilium. And these mice completely lack the outer segment seen in these wild type retinas, and they're completely lacking the transition zone. So nearly all the cilia types in the mammalian body are affected by loss of TMM67 cleavage. So the other really important thing about TMM67 that's been published by many groups is that its N-terminus has this cysteine-rich domain, which is highly homologous to frizzled family of cysteine-rich domains which act on Wnt signaling. And it's been shown that um, TMEM67 makes a complex with Wnt5a and ROR2 and is involved in non-canonical Wnt signaling. So the first thing we wanted to look at was, since ADAMTS9 cuts in between this CRD and the BRD, what does this do to this pathway? So we first confirmed that indeed what was published previously by other groups is true. So you can look at ROR2 phosphorylation by mobility shift assays. These are wild-type MEFs treated with Wnt5a. And when you do that, ROR2 gets phosphorylated and migrates slower, and that's why you see this shift. And TMEM67 knockout MEMs do not respond to Wnt5a at all, and they do not show an increase in this molecular weight. To our great surprise, the delta cli MEFs respond to Wnt5a very similar to the wild type. And more interestingly, when you look at the brain, the TMEM67 knockout ependymal cells have this massive upregulation or increased level of active beta-catenin staining. And the delta cli mice, this signaling path is completely normal. So what we published last year was that ADAMTS9 mediated TMEM67 cleavage regulates or is essential for two functional forms of TMEM67. It generates a C-terminal fragment that localizes to the ciliary transition zone and is involved in ciliary transition zone assembly, and the full-length form involved in the non-canonical Wnt signaling, which also regulate the canonical Wnt signaling pathway. Last year, when we were doing these experiments or publishing this, we didn't really know what the function of this small N-terminal fragment that is being released, or whether it has anything to do with Wnt signaling. But Manu has been very busy since publishing this work, and now I can show you that this fragment is indeed bioactive. So he has generated stable cell lines expressing just the N-terminal fragment, which we refer to as N331. And this is made in L cells, same as the L cells expressing Wnt5a that is being widely used in the Wnt signaling field. And if you treat MEFs with the conditioned media overexpressing this fragment, you can see here, using phospho-junk signaling and nuclear localization, this can activate the non-canonical Wnt signaling pathway. So this has a long-range signaling ability as well, and we're really, really excited about this finding. And if you combine, we have a cell line that expresses both Wnt5a and N331, and that has the highest activity in terms of phospho-junk activation. So we need to update our model from last year. It's not two functional forms. We now believe there are three functional forms that are being created by this simple cleavage of TMEM67. And more importantly, we find this fragment also present in the cerebrospinal fluid. So we're trying to figure out if this fragment can act as a signaling molecule in terms of what's happening in the brain. But what about the kidneys and ADAMTS9? If you delete ADAMTS9 in mice, this is what we get. This is 1 week post weaning. This is ADAMTS9 conditionally knockout male mouse. They started moving like this with weird gating, and they become much smaller compared to the wild-type littermates. Female mice, on the other hand, are completely normal at this age. There are two conditional knockout females, which we cannot distinguish at this age. So this is using 63, and we're deleting ADAMTS9 in the proximal nephron, and the reason for this is that it's very much restricted to the proximal tubules and the glomeruli. And what's really interesting is that both ADAMTS9 and TMEM67 are highly expressed by the male proximal nephrons compared to the females. And this is using the Kidney Cell Explorer published by the McMahon lab. and 7 and 8 refers to segment 3 of the proximal tubules. So the phenotype is very sex dependent. So remember, we are deleting embryonically, and this is at 21 days of age. ADAMTS9 conditional deletion in males produce these highly cystic kidneys, but they are quite small compared to the wild type. To delete IFD88 using the same Cree line, these generate much larger cystic kidneys, and they don't survive till P21. The IFD88, the maximum we can get are 2 weeks by this Cree, and at this age also they're much larger. And in comparison, this is how the TMM67 knockout looks like. So these are all male kidneys. Females feels like they last forever. They become highly cystic at about 12 weeks of age. We really don't know much about why there is this sex discrepancy between males and females, other than that ADAMTS9 is highly expressed by the male proximal tubules. I'm going to go through this data quite fast. Essentially, what I want to show is that compared to, let's say, IFD88 or the TMM67 knockout, the ADAMTS9 conditional knockout kidneys are much smaller, and they do not increase in size, similar to the IFD88 and the TMM67 knockout. So when does cyst development happen in these? At the end of embryonic development at E18.5, the ADAMTS9 conditional knockout kidneys, both male and females, look quite normal. We don't really see much cysts at this stage by H&E staining. You can see few cysts in the IFD88 and the TMM67 knockout. It's at P7 we first see cystic expansion in these kidneys, and both male and female kidneys are cystic. And for whatever reason, after the first week, the females slow down quite dramatically and they survive, but the males continue to become highly cystic and die around 21, 28 days. And another point is IFD88, for example, started enlarging at this point as well, which was quite obvious. When you do LTL and DBS staining, although we couldn't see cystic expansion, we could see that the proximal tubules are not quite normal in the ADAMTS9, and that's what's shown in this box here. You can see these beautiful proximal tubules. In the wild type, these in the mutant, they started to enlarge at this stage as well. In terms of the number of glomeruli and the kidney area, at E18.5, essentially all 3 of the models are pretty much normal compared to wild type, and the number of glomeruli is not decreased in the ADAMTS9 either. So we don't think embryonic kidney development was affected by loss of ADAMTS9. It's postnatal cystic expansion that is affected in the ADAMTS9 conditional knockouts. What about celiogenesis? So this is freeze fracture SCM, looking at male cystic kidneys in this particular example. And in the loss of ADAMTS9, you do not see any primary cilia in these cystic lumens. Whatever cilia that you see are really, really short, marked by these red arrowheads here. We did TM as well, and we've identified few of these really short cilia, and these lack the ciliary transition zone that I mentioned previously as well. So you can look at this using immunostaining as well. These are the wild type kidneys on the top, marked by a IL-13B and FOP. You can figure out these beautiful primary cilia. They are completely missing in the ADAMTS9 conditional knockout. So, as I mentioned, loss of either TMM67 or ADAMTS9 in RPE1 cells caused this loss of this MKSP9 module, and we wanted to see if this is true in renal primary cilia as well, and that's what Sydney did. We looked at very particular for these 6 molecules that we know that are affected in human cells. And what she found out was that everything other than CC2D2A is lost in renal primary cilia as well. These are renal primary cultures of wild type and ADAMTS9 conditional knockout kidneys. And we looked at the extracellular matrix on this as well. And ADAMTS9 conditional knockout kidneys have this massive expansion of collagen or deposition of collagen, which is shown by Mason trichrome staining on the top row and by collagen 4 immunostaining in the bottom. Collagen 4 is pretty much limited to the glomeruli in wild type. And you can see here in the ADAMTS9 CKOs is throughout the kidney in these CKOs. Now, I want to show one more extracellular matrix molecules because this is differentially regulated compared to the other models. That's laminin alpha 5. This is a molecule that's identified by Jeff Miner's group and shown that when you knock out laminin alpha 5 in mice, this also leads to polycystic kidney disease. So we want to know what happens to laminin alpha 5 as well. In the ADAMTS9 glomeruli, you lose this laminin fibril formation. And the staining pattern looks like this. In the right-hand panels, we are showing TEM, characterization of the GBM. In the ADAMTS9 CKOs, you see this very thick GBM. It's somewhat lighter colored compared to the very well organized GBM in wild types. And we also know that there is a massive amount of renal fibroblast marked by MES 123 antibody staining that are present in the ADAMTS9 CKO kidneys. And these are also highly fibrotic, shown here by alpha SMA staining. So we believe these changes are occurring through gene transcription, and we are characterizing ADAMTS9, IFT88, and the TMEM67 knockout rules here. What we're seeing is TGF beta signaling is highly upregulated, which is a master regulator of extracellular matrix. And we also see upregulation of these few hand-selected extracellular matrix molecules, including fibronectin and collagen and hyaluronan synthase. Now, very specifically, laminin alpha 5 transcription is not affected in the ADAMTS9 conditional knockouts, but it is upregulated in IFT88 and also in the TMEM67 knockout. And this is smooth muscle actin. HAVCR1 is a SCIM1. This is a kidney injury marker. It's all upregulated in all three models. Let me go back to laminin alpha 5 and just show you the difference. In IFT88 conditional knockouts, this is highly upregulated, and you can see blazing amounts of laminin alpha 5 in the GPM. Collagen, on the other hand, is not very much different compared to a wild type level. There is a little bit of collagen 4, but nothing compared to the ADAMTS9 CKOs. And the IFT88s are also highly fibrotic, shown here by alpha SMA staining. So what about TMEM67 cleavage? Is this affected? And we looked at this using the primary culture. So if you look at the conditioned media of ADAMTS9 conditional knockout primary cultures, there's a decrease of this N-terminal fragment, this 34 kilodalton fragment that's generated by ADAMTS9 mediated cleavage. In our delta Cli model primary cultures, this fragment is completely gone, and you can see increased levels of the full length TMEM67 molecule as well. But very specifically, there's a difference between ADAMTS9 and the delta Cli models. The delta Cli kidneys are highly enlarged compared to ADAMTS9. As I mentioned, the delta Clis have these long or short cilia. We looked at this by immunostaining, and we used IL-13B. And what we found out was that these really long cilia not normal in terms of IL-13B trafficking as well. So what Sydney's showing here is that the really long ones have less intense IL-13B, and the really short cilia has very intense IL-13B localization. And very interestingly, in our TMM67 models, we also see a lot of these nuclear localized IL-13B as well. This particular antibody that we're using is against the C-terminal domain of IL-13B, and it's been shown that if you GFP tag the C-terminal domain of IL-13B, there is nuclear localization. So this is very unique to TMM67 knockout. We don't see this in the ADAMTS9 or the IFT88, for example. We don't really know why there is this localization, but we decided to show it because it's highly reproducible. So we characterize the transition zone assembly as well. I'm not going to go through all the data, but essentially similar to the ADAMTS9 CKOs, all 5 molecules, TCTN1, 2, 3, TMM237, and B92 are significantly decreased in the renal primary cilia of TMM60 knockout and the delta clee models. For whatever reason, CC2D2A is not affected in renal primary cilia in the loss of ADAMTS9 cleavage, but it is affected in RP1 cells. So I mentioned there are 8 total mutant alleles that are known for ADAMTS9 now, and we wanted to know if these are defective in TMM67 cleavage as well. We first characterized their cytogenesis ability. So these are wild-type RP1 cells on the very top left, transfected with an empty vector. The ADAMTS9 knockout cells have very few ciliated cells. If you transfect full-length wild-type ADAMTS9, you can rescue ciliogenesis, but our ciliopathy patient variants do not rescue ciliogenesis, and both number of cilia and the cilia length are affected in these ciliopathy patient variants. The only molecule or the only variant which is in the very C terminus of this enzyme, this S1826N, this is in the very C terminus of ADAMTS9, that has a partial rescue of cilium length. So this is far away from the catalytic domain, and this seems to be able to function to a certain degree. This is looking at TMM67 C terminus localizing to the transition zone, and all these patient variants are defective in TMM67 localization. So we believe the ADAMTS9 patient variants are really important in regulating TMM67 transition zone assembly. Now, as I mentioned, TMM67 plays an important role in Wnt signaling. So we wanted to look at this as well. And what we found out was that ADAMTS9, IFT88, or TMM67 complete deletion, all these 3 cystic kidney models have highly upregulated canonical Wnt signaling. This is looking at active beta-catenin staining, and that is not happening in the delta clee models. For whatever reason, the delta clee model, which has the full-length TMM67 molecule at high levels on the cell surface, can block this upregulation of canonical Wnt signaling, and this is one of the major differences between these 3 different models. And we believe this is happening through upregulation of non-canonical Wnt signaling target genes. So BLOT2, LCOR, and HADH, these are known downstream target genes of canonical Wnt signaling. Neither the ADAMTS9, the TMM67 knockout, or the IFT88 show a clear directionality in terms of what happens to these molecules in transcription. But our TMM67 delta clee mice show a massive upregulation of these known target genes. So we think it's this downstream signaling pathway that's somehow blocking the upregulation of the canonical Wnt signaling pathway. So one of the major questions at this point we had was, why are the ADAMTS9 kidneys small? And we believe it's because of its ability to regulate the collagen levels in the kidneys. So these are Mason trichrome staining, looking at low magnification images here. And the ADAMTS9 male and female cystic kidneys have massive amounts of collagen compared to either the IFT88, the TMM67 knockout, or the delta clee models. And this, we think, is the major difference between these models. And why is the collagen level completely different in the cystic kidneys? And we got PKD1 and PKD2 conditional knockout kidneys from PAM. And this is how the Mason trichrome staining looks like compared to the ADAMTS9. These were all done at the same time, and we don't see a massive amounts of collagen compared to ADAMTS9 in these kidneys. And collagen 4 by immunostaining is not dramatically increased either. So this is where this other molecule that we discovered comes in, and we believe the increased collagen level is due to the ability of ADAMTS9 to cut MT1-MMP. So we first published this actually before we even started working on TMM67, and what we showed was that ADAMTS9 cuts MT1-MMP, which is one of the major metalloproteinases in our body that's regulating collagen levels, and it creates this soluble form of MT1-MMP. And this cleavage is in between the catalytic domain and the hemopexin domain. Now, MT1-MMP is also important in activating MMP2. So this is happening at the cell surface. MT1-MMP removes this pro-domain of MMP2 and activates MMP2, which is another collagenase. And we showed that ADAMTS9 knockout cells have increased the level of active MMP2. So there's a ton of this on the cell surface. And we also use DQ collagen 1 to look at collagen cleavage at the cell surface as well. And these have increased DQ collagen signal as well. So we believe the reason why the ADAMTS9 model is different to the others is because these have high levels of interstitial collagen that is regulated through this cascade of events mediated by MT1 MMP cleavage of ADAMTS9. And we believe that's what's controlling these large ADP-KD-like kidneys versus small NP-HP-like kidneys, where the interstitial collagen acts as a barrier for expansion. So this is our working hypothesis at the moment. We don't see this level of collagen in any of the 5 cystic kidney models we've looked at. And this is highly unique to ADAMTS9 cystic kidneys. It's essentially covered in this sea of blue or collagen. And we believe this is what's preventing it from expanding, and it models the human condition quite beautifully in this case. So just to summarize what I showed you today, we looked at 4 different cystic kidney models related to ADAMTS9 mediated cleavage. We interfered with this pathway in 4 different levels, either by losing ADAMTS9 or by TMM67 knockout model or by the delta cleave model, which does not have this C-terminal fragment or this N-terminal fragment. And some of these extracellular matrix changes are mediated through both Wnt signaling and downstream signaling events that are happening downstream of the primary cilium. So transcriptional regulation is important here as well. But just to summarize, the 4 different models we looked at, the main difference we see is that the ADAMTS9 conditional knockout is very severe in males and less severe in females. The females take a much longer time to develop cystic kidneys. The males die around 21 and 28 days. In the ADAMTS9, the TMM67 knockout and the IFT88 conditional knockouts, canonical Wnt signaling is highly upregulated. And in the delta cleave model, the canonical signaling is not affected, but they have highly increased non-canonical signaling pathway. I didn't really talk about the immune response in this for the lack of time, but there is a high degree of immune activity in the ADAMTS9 conditional knockout as well. I'm happy to talk about this and show some data at the end if any of you have any questions. But we believe this increased level of ECM or collagen is what's regulating both the immune response and preventing cystic enlargement of these kidneys. So I promised you I will come back to this work of art. What was lacking in this original picture was light, and this is our current way of thinking, and there's the motorbike, as I promised. So I don't know how long this artist took to generate this particular piece of art using 848 pieces. But for us, it took almost a decade to figure out how NMDS9 regulate cytogenesis and also Wnt signaling. So with that, I'll thank the people who did the work. Most of the work related to TMM67 cleavage was done by Manu Ahmed, who's a postdoc in the lab. And the new work on NMDS9 conditional deletion was done by Sidney Fisher, who's a graduate student in the lab. And the N-terminal fragment work is also spearheaded by Manu, and we are currently working on getting that work out in bioRxiv as well. And I need to thank Karen Lang and Oliver Black for the C. elegans work, and Colin Johnson for the TMM67 reagents, and obviously Frida Hildebrand for discovering the ADAMTS9 initial ciliopathy patient variants, which essentially opened up a new paradigm into this metalloproteinase, which we previously thought was just cutting extracellular matrix molecules, and also to PAMTRAN for giving us the PKD1 cystic kidneys. With that, I'll stop and take any questions you have.
Thank you very much, Samita, for sharing your beautiful work with us. So I saw in the chat that there are a couple of questions. So one of them was, what cells are these? And I think this was asked sometime early during your talk. I think it might have been RPE1 cells. Did you use RPE1 cells at some point?
Yeah, the initial transition zone characterization was done on TMM67 knockout and ADAMTS9 knockout RP1 cells. In Sydney's paper, we also look at renal primary cultures as well. So, and we just confirmed the 6 molecules that we identified in RP1 cells in terms of the transition zone were also affected in the renal primary cultures as well. So everything other than CC2D2A was affected. I'm not sure what side the question was on. I'm sorry.
Okay. Brad has a question.
Wonderful work. Really curious on the difference between the males and the females that you see. And whether that's, if you look in the females, which I believe were protected, right? I can't remember which way it went, but are the cilia defects still there?
Yeah, it's there. So what's happening is that they start the female, so it's more severe on males and less severe on females. So I think this is somewhat similar to, with the PKD1 and PKD2 late onset model. My hypothesis is that something happens to the female cystic kidneys that prevent them from becoming further cystic or expanding. So I don't know if it's the hormones kicking in that are female specific that are protecting the female cystic kidneys, or if ADAMTS9 has some sort of kidney injury response that is more specific in the males. We haven't really figured it out, to be honest. And the male and female specific ADAMTS9 localization, the transcript levels that I showed you from the single cell RNA-seq, that's on adult mouse kidneys, right? So we don't know when that switch happens, if the increased or the prolonged expression in males occur and the females drop out. So it could be both of, any one of those pathways. We really haven't figured out why there is a discrepancy.
I guess you haven't crossed these with like the PKD1s or 2s to see if it rescues or delays cyst formation in that background in one sex versus the other.
We have not done that.
Okay, Martin has a question.
Oh, yes, great talk. And I liked your theory about the collagen preventing the cyst expansion. And I thought one way you could test that hypothesis is to look at the difference between the cysts that are on the surface versus the cysts that are on the interior of the kidney. Because in the humans, those surface cysts become exophytic and huge because they're not being constrained by any parenchyma. And here your surface cysts, you would think if the collagen is preventing cyst growth, then those ones on the surface would be less affected.
Right. Yeah, that's actually an interesting point. We can definitely look into that. See if the size of the cysts are different from parenchyma versus internal. But again, we are deleting ADAMTS9 in the proximal nephrons very specifically, so it's not a global deletion we are doing in this. But yeah, we can definitely look into that, and that's a interesting idea.
Oh, Chris, would you like to ask your question?
Hey, thank you. This is Christian, pediatric nephrology. Thank you for the talk. Question is the degree of fibrosis in these subjects compared to other ciliopathy translate into more prominent anemia compared to other ciliopathy mice model or patients? Because we know in the nephrologist you will have the degree of anemia is out of proportionate degree of your GFR reduction. The theory is they have more fibrosis. So have you noticed that in these mouse model?
We haven't looked at that, and that's, I think, a very good point as well. And I think, a case study or, the patient variants are published by many different groups. So I think it would be interesting to look at this in the clinical setting as well. But in mice, we have not looked at this particular question.
Thank you so much.
Fang?
Yeah, hi. Great talk. Here's from Chen University of Maryland. Yeah, I have a basic question. This Adam TS9, this is a matrix protease,
Yes.
right?
Yes.
So in the context of a tubule, I would imagine that the sitting at the basement membrane, so is the basal side of the tubule.
Right.
So if we cut TM67 and releasing the C-terminal tail, which go to the cilium, go to the transition zone of the cilium, right?
Right.
So, but the cilium is the apical. So how is this two topologically, how are they connected? One is acting at the basal body, basal side of the tubule. The other one is at the apical.
Let me jump to, so did I stop sharing the screen, I guess? So let me jump to this model again. So you're absolutely right. So, it's I believe the internalized protein that's important for the ciliary function. So, this can happen either at the apical surface or the basal surface, right? So if you think of a renal epithelial cell, you would think, all right, this needs to be secreted not from the apical surface and from the basal surface, but I would argue that it's these endocytic recycling endocytic vesicles that are important for the ciliary function in that context. So once it's internalized, it can be trafficked to the cilium through that.
So you're saying that it internalized Adam TS9
Right.
actually go to the basal, I mean the apical side, and doing all
Right.
this cutting to the TM67.
Right.
Yeah, that makes sense.
And I need to point out, this is never part of the vesicle membrane. Okay, so this is part of the lumen that is being. It's a secreted molecule. It's once it reaches the cilium, it's released from the base. And if I go to the very last slide, actually, this is Adam TS9 on my acknowledgement slide that I had here as well. So it's highly enriched in the surrounding the cilium as well. One more slide. So this is actually ADAMTS9 in red in these vesicles that I mentioned to you in this particular image. So it's once these vesicles fuse, it's released and it's never found inside the cilium itself. So it's in the lumen of these vesicles.
So when you look at this, is this a kidney tubule? Is the kidney IF the one?
No, this one is a cultured RP1 cell.
Okay. Well, have you looked at the kidney itself? Where is those vesicle endosome?
We have not. We have not done this staining in primary cultures, for example.
Mm-hmm.
Yeah.
That would be interesting to see it.
Yeah, I completely agree.
Mm.
But once you culture it, though, you don't get that true, apical basal polarity. So you need to do it in a 3D model, I would say, or some sort of 3D ogling model or tubular model.
Okay, so the hour is late, so there's just two quick questions left in the chat, Sumeda. So one is, did you see fibrosis in other organs like the liver?
So I'll let me see if I can show you the liver. These macrophage. I can put the liver work somewhere here. Okay, so it's not fibrosis per se that we're seeing, although we see at least, so this is TMM67 delta clean model, okay? They have a liver phenotype, but it's not fibrosis. What we're seeing is an impairment of hepatocyte differentiation and the branching morphogenesis of the vein. So the hepatic portal vein here seen in the wild type, which is completely missing in the delta clees. So I know TMM67 is very important for liver function as well in human patients. This is what we're seeing in our model. There is some increased collagen, you can argue, by meson trichrome staining, but the more profound effect is, you can see at 11 how the kidneys look, the liver looks different in these. In mice, it's mainly a branching morphogenesis defect that we are seeing in the liver, unfortunately. And ADAMTS9 is not expressed in the liver as far as we know, so it's not clear which enzyme is cutting TMM67 in the liver. For example, in the brain, we know it's the sister protease ADAMTS20 that's doing it.
Okay, thank you. I also see there's no cysts in the liver as well. So final question, did you have a chance to see the status of ADAMTS9 or delta clee or TMEM67 in PKD1 or in NEC8 mice?
We have. I can tell you both PKD1 and PKD2 are downregulated in ADAMTS9 CKOs. I can't remember what happens to ADAMTS9 in the PKD1 and PKD2. We've done that analysis. I just don't want to give you a false answer. We've run PKD1 and PKD2 levels. I just can't remember what direction it goes. I think it's decreased. I'm not 100% sure, but we have done that analysis, at least by qRT-PCR. What we do not know, but we would love to know, is if PKD1 and PKD2 ciliary localization is affected by the loss of the transition zone. We haven't found a good antibody that works in mice. I really want to know if, in addition to IL-13P, if PKD1 is also mislocalized in this really long cilia we are seeing in this model.
I think there are some good PC2 antibodies, and maybe Feng has some PC1 antibodies. I don't know, Feng, if you can vouch for the antibodies.
Yeah, and he works in immunostaining, but yeah, I'm open to trying them.
Okay, so I think our final question always for our guests is there anything that the PKD-RC can do for you in terms of reagents?
Yeah, so that's a good question. So let me get back to this. So there is some work done by Garcia Gonzalez lab and looking at MMP inhibition. This is a pharmacological inhibitor of that's known as mermistat. And I'm really interested in looking at the collagen level differences in the PKD1 versus nephrocolitis models. So in that sense, I'm really interested in getting this PKD mouse lines and doing interminomics to characterize what happens to these different extracellular matrix metalloproteinases and how they regulate the extracellular matrix by proteolytic cleavage. In PKD models. But why this data is interesting is that if you inhibit matrix metalloproteinases, what you get is this really small but highly cystic kidney. So I really do think that these are highly involved in regulating cystic kidney size, based on this preliminary work done by others.
Right. Okay. Well, yeah, you can definitely get these mice from the PKD-RIC site, Sumita. Just wanted to thank you so much again for a really terrific talk and for sharing your wonderful work. Thank you to everybody who joined today, and we'll see you in a month.
Thank you for having me. It was a pleasure presenting.